Now take questions for both of the speakers. Jennifer, you can come up, and Patricia, stay on the line, and we'll take speakers from the audience. Remember to use the microphone. So, question in the back.
Yeah, Jennifer, so you have a very nice comparative method examples there. And so you have a lot of instances I could see in some of your trees where you had the same trait evolve many times along the tree. And I don't know how much is known about these individual traits, but there's probably genomes for at least one or two of all of those clades. So I'm wondering if you'd thought of taking, say, okay, I have many independent evolutionary to pH or whatever it is that you're interested in, and then looking to see, since you have so many independent evolutionary events, what happens each time in those genomes that would be, you know, what is the response and why do they have those traits? Because you have so many independent ones, you could ask, what in the genome is responsible for those traits? Have you thought of doing that?
Yeah, I haven't done it myself, but I believe Stuart Jones, who's one of my co-authors, has a very nice paper out where he was looking at that in response to something where they were looking across samples, actually at the whole metagenomes, and trying to pull out, well, what are the sort of genomic indicators of changes. And so you might be able to do that also at a genome level as well. I think that's a great idea. Yeah, you could certainly start doing that to try to better pinpoint what's going on there. Yeah.
A question for Patricia. So it seems like a very interesting and potentially helpful model. I have a question about one of the details. I assume that you're modeling the impacts of increased indoor relative humidity from tightening homes and other kinds of interventions, and somehow connecting that to risks from dampness and mold. But what I'm curious about is the risks of mold growth are related more closely to the, say, the moisture content or water activity of the materials in the house, and much less to the relative humidity in the home. So how do you make that connection?
So for this model that we built, and we published it a couple years ago, so it's a little bit outdated. So you're right, the absolute humidity level is more predictive of growth and even survival, right, through the influenza as well, than relative humidity.
Well, I wasn't saying absolute.
And what I did is I drew from the literature, the building literature, that has these isotherms looking at how long basically it's building literature, putting pieces of drywall, pieces of wood, pieces of different building materials in controlled chambers, and then measuring relative humidity, temperature, and then building these curves, and then saying relative humidity has to be this level for so much time,
right,
and the temperature has to be this, et cetera. And so we created a series of differential equations that depended on both the humidity and the temperature to predict the probability of whether there was going to be mold or not. Now, I know Tina Raponen, after we published this, published on the ERMI index, and I think that that's probably a better way to go, but we have not updated our model to do that.
Thank you.
More questions? Over here.
I have a question.
I'm sorry.
What are, Jen, what are your thoughts on using 16S for identifying geographic variation in bacteria or traits, like things you were talking about?
Yeah, so I think what I think our work is getting at is that if you are using 16S or any other marker for taxonomy, that the type of variation that you're actually going to be seeing is really, really, so the type of variation you're going to be seeing and capturing is going to vary. So if you use 16S, you can only go so fine because it's a very conserved gene, and so you're not going to be able to pick up variation in 16S that's for traits that are not well conserved, right? So the standard these days for classifying 16S into OTUs is 97% sequence similarity of a very small section of the gene. And the back of the envelope calculations that I know of, that would put your taxa diverging at about 200 million years ago. So all the evolution that may have happened within the last 200 million years is not going to be captured by that variation. So it just depends on what kind of variation you're looking at. If you're looking at traits that evolved earlier than that and are still reflected in the vertical taxonomy, the vertical inheritance, then that will be fine. So yeah, it's just something to keep in mind when you're doing that.
So this question is for Patricia. So you have mentioned in your metadata about viruses and fungi, but what about the bacteria and its influence on those, healthy effect of the occupants?
On the healthy effect of the what, sorry?
The people living inside the home. You mentioned a lot about the fungi and virus with asthma, but we haven't heard anything about bacterial composition of those built-in environment and its effect.
So that's an interesting question that we've thought about, but I don't know that there is a lot of epi-literature linking bacterial concentrations to asthma attacks in a way that we could incorporate into the model. So we needed to have, in order to be able to do this, right, you have to have a way to model exposure, so emissions somehow, and then you have to have the linkages between exposure to certain numbers of bacteria or types or species of bacteria and how that impacts lung function. You need to tie it to a specific lung function. In our case, we found enough information to link it to FEV1% predicted, which is the amount of air that you can breathe out in 1 second if you're trying as hard as you can. And then, that lung function to the probability of having an asthma attack. And in order to incorporate any of these things into the model, you have to have all those linkages. And I don't think, though, it exists for bacteria.
Hi, this is Bob Holt. I enjoyed both your talks. I have a question for Jennifer, which is you have this, you called it a sort of a hierarchy of traits in terms of phylogenetic conservatism. It seems to me you could do experimental evolution studies with the bacteria, and you have an a priori prediction about which traits you can relatively easily select on in the laboratory, and which ones is just tough. If you try to select on them, they just go extinct or something like that.
I think that's right. I mean, you could think about those sorts of experiments both in terms of the whole community. You could put a whole community in soil and expose it to some perturbation, and we'd be able to look at sort of what genetic resolution we're shifting. But you can, of course, many people have evolved bacteria, for instance, to different temperatures. And I don't know if you noticed, I was surprised that the temperature trait was actually relatively less conserved. I had sort of thought of that being a trait very deeply conserved. And I think what's going on there is that, whether or not you're a psychrophile or a thermophile might be very deeply conserved at those extremes. But we know from lots of lab experiments that you can evolve E. coli to shift its optimal temperature by 5 degrees with no problem in only hundreds of generations. So there may be some range within maybe the plasticity of the organism that you can evolve preferences pretty quickly, but then at a more fundamental level it would be broader. And I think that would be very interesting to explore with these sorts of experiments.
So Patricia, I have a question for you from one of our remote committee members, and she asks, is the CONTAM pollutant simulation to study particulates and airflow markers really tested specifically related to good versus bad microbials?
No, I don't think. I think that the way that it's been used has just been looking at particle sizes. So for example, the influenza work that we did was looking at emissions from people, what we knew about what particle sizes they coughed or breathed, and then modeling that as particles. It would be the same approach for modeling things like mold spores, et cetera. So in as much as biologicals would have the same behavior as a pollutant that you would find, another pollutant that you would find in the air, then it's the same modeling principles, right? It moves with air streams. There's air exchange rates for fresh air, et cetera. I don't know that it's that I know of. I could be wrong. That there's been a lot done with contam and biologicals.
Let me answer that because we've worked a lot with contam and biologicals in my group. You know, fundamentally the physics is the same. The only significant differences are the degree to which you might need to introduce terms associated with sticking of the biological particles to surfaces and rebound. But fundamentally everything else is the same.
For survival, maybe would be another thing to think about.
I didn't catch that.
I was just thinking survival might be another parameter.
Oh, yes,
that you
yes, certainly, you know, but that's a
indicator.
reaction term. Yep.
Mm-hmm.
More questions? Okay. What's our instructions?
Break?
Break for 15 minutes?
Thank you.
Speakers?